Okay. So today we're starting the first of 3 sessions on single-cell RNA-seq. So today I'm going to be, good, move this down. Less, can you hear? Less, maybe. Let's see. I'm not sure what I want to do to. Thanks. Can you turn it down like a little, Otis? Oh, no, okay. Okay. Can people hear? How's the echoiness? I think it's okay. It's not bothering me. Can everyone hear? Can people on Zoom hear? I'm hoping your silence means yes, you can definitely hear.
Yes, we can hear.
Okay, cool, thanks. Okay, so this is the other thing. This is the first of three sessions that we're doing on single-cell RNA-seq. So today I'm going to be giving kind of background in theory, explaining really how the technology works, and like very sort of at a high level talking about how we start to analyze it. And at the towards the end, we are going to do very brief hands-on with running the bioinformatics pipeline, and I'll talk more about that later. And if there's time, we're going to kind of walk through a little bit of a toy example. If there's time, I'll sort of show you a little bit of a more realistic way for doing the bioinformatics. And then for the next 2 weeks, Tyler Shapey is going to be talking about more of the sort of downstream analysis, both like the statistical analysis and data visualization sort of things. And I think that's all I have to say to you, except that, so like I said, we're going to start off, we are going to get this running. So I'm going to leave this up for a second. And then I'll actually show you how to start things up. So we're going to be working through DCC on demand as usual. The one thing that's going to be different is that we're going to be asking for more CPUs and more memory, because, like I said, this is already going to take an hour and 15 minutes to run, and we go for less CPUs, but it would take even longer. And I haven't really played too much with the memory limits. I think they're recommended. I think 64 might be the recommended minimum. Okay, anyone need me to leave these links up longer before I switch to actually walking through? Okay. So let me do, let's see if this is still signed in. Of course it's not. Okay, which way am I going? This way. Okay, so you should go to DCC on demand. Anyone not here? Cool. Okay, and you want to go to interactive sessions. And as usual, and then go to, CHSI, RStudio Workshop. And like I said, so 4 hours should be plenty of time. We're going to change the CPUs to 16, which I think is the maximum that this app allows. And we're going to change the memory to 64. And then everything else should be totally fine. And so I'm going to, anyone need me to go through that again or go questions before I launch? Okay, I'll launch. Do I need it to be bigger? Anyone want me to make things bigger? It's fine. Okay. Okay, so that got started nicely. We're gonna click on connect to RStudio server. I don't know what that's saying. We'll ignore it. I'm going to switch to, actually, I can't remember if I have, well, I guess we'll find out. I'm going to open the project. If you don't, what am I looking at? There. Yeah, this, right? Okay. So if you don't have the project open, you should open it. And there we go. Come on. Thinking. Okay. And I'm going to pull, probably if you haven't already done a pull today, then you should do that too. Go back and show to start again. Yep. Where do you want me to start from?
Just to get the, I think I don't know where to get this folder. So once R is launched, then where should I go?
Pull is being aborted. So go to, once RStudio is launched, is that what you said?
Yes, sir.
Okay. Have you cloned the project before? The repo? Like, were you, let me think.
Yes.
2 weeks ago, I think I remember you were in the session 2 weeks ago. Did you work on the, do the hands-on stuff then?
Yes.
Okay. So then you should be able to go to, so if right here, it says, actually, maybe let me make this a little bit bigger. So if right here it says 2023-2024 HIV workshop, then you should be, you're most of the way there. Does it say that there?
Yeah, this I don't see exactly the same one. It's RNA-seq part 1. So maybe it's
For here?
yeah.
Huh. Okay. So go to okay. So one option is if you click here and do you see in any of the options here 2023-2024 HIV workshop?
Yes, yes.
Okay, so click on that.
Yeah.
And then it should it'll spin for a minute.
Yeah. So yeah, workshop series. I think I see that one now.
Okay,
so it's 2020 workshop series are. Yeah, I see that one.
Okay. And so then you should be able to go to click here where it says pull and click on that. And hopefully it'll work. Oh, I'm getting the same issue. Okay, so this is the same issue that Elizabeth had. Okay, so what if you're having this problem, then maybe I'm getting a slightly different. So I'm going to fix my problem, and we'll see if it helps people, if it helps Elizabeth or anyone else.
I had this problem. I deleted the ignore time and then it would.
Yep. Yeah. So if you go here to where it says .getIgnore, if you click on the checkbox next to it and click on delete, and it'll say, are you sure you want to permanently delete .getIgnore? I'm going to say yes. And then I'm going to click on pull again. Sorry, this might be my fault because, and so now now the pull worked. So Elizabeth, can you try that? And the 11 commits, but these are not worth sharing. Is there a way to ignore these? Yes, there is a way to ignore commits. I'm going to check my notes because it's something that I'm pretty sure I know what we want to do, but
it's okay. I got it to work using the first method.
Oh, it worked? Okay,
yeah.
Great. Cool. Okay, so has anyone not been able to do a git pull?
It says create mode something, and I don't know if it worked or not. So
if you do a pull and it works, it doesn't really, it'll say create mode something, and it doesn't say exactly it worked, but it won't give you an error message. And it should, that's another good way. Oh, okay. One way that you can check and see if it worked is if you, come on, I don't know why the cursor's being laggy. If you go here on this little clock and click on that, nothing's happening. There, oh, whoops. Yeah, that's why. Okay. So if you look, this will show the history, and it should show at the very top of the history, it should look just like mine, where it says in green, it says head, points to ref, head, main, and then it says, so it's like this. The most important thing you want to look at is if it says assay toc.md here, and then if this sequence of letters and numbers match up with mine, where it says 8BE3D084, then you're all set. Okay, good. Anyone else having an issue? Cool. Okay, so what we want to do is, I have to think for a second. We want to, I'm just going to close all this other stuff that I have open. I don't know why I have it open. Oh, I know why, because it's open from the last time I taught. You don't need to close anything if you don't want. Okay, so we're going to go in the assay, go down here in the file browser in the assays directory and click on that, and then click on single cell RNA-seq, and then click on, I'm going to make this a little bit bigger. And I'll remind you, if you kind of miss a step here, then you can look right here where it says, this will tell you exactly where I am. Okay, so we're in single cell RNA-seq, and we want to go to notebooks. And then there should be three files in there. You want 03 day 4 cell ranger demo.rmd. So click on that. Anyone not here? Okay, and then what we need to do is just click on, I'm going to click on the little arrow next to where it says knit, and then click on knit to HTML. And hopefully if I didn't screw anything up, then that should start working. You should see a little tab here that says render, and it's spinning. And so, like I said, that should take about an hour and 15 minutes. Hopefully it's not slower because a bunch of us are doing it at the same time. And we'll come back to that. Anyone not have that running?
I have error named the input file cannot contain special shell characters, something. When I do knit HTML, when I just click on knit, it works fine.
That's not a good sign because it shouldn't have finished so quickly. Now the next question is, is mine? I'm getting something different. Oh, maybe it's because I didn't clean up. Okay, hold on. Let me clean up really quick. Okay, tell me what yours says again.
Knit to HTML, giving me an immediate error. What characters are involved in it? Oh,
Solved it? Maybe? Okay, hold on. I'm going to clean up my mess. Oh, good. I like those problems. I just have to solve my problem. I'm assuming everyone else is, come on. Come on. Okay, I'm assuming this is going to be done in a second. Everyone else's is actually still knitting, right? It's just me that's having a... Okay, I'm going to knit that again. And I hope that it actually works this time. And I'm going to go back to my slides. Okay, so everyone got that going okay, right? You'll yell if it didn't work. Okay. So now, like I said, we'll talk about what that actually was about in hour and a half, 2 hours, something like that. So what are we talking about today? So 2 weeks ago, 3 weeks ago, when I talked about RNA-seq, I told you that you know, depending on how you want to think about it, there are different ways of kind of grouping RNA-seq experiments. And so one way is to think of, you know, put one class of experiment in the bulk RNA-seq bin and the other class of experiment in the single cell RNA-seq bin. And last time I talked 2 weeks ago, and last week you talked about bulk RNA-seq, and today we are going to be, and like I said, for the next 3 weeks, next 2 weeks, after today, we're going to be talking about single-cell RNA-seq. So I want to give you a little bit of an intuition in, maybe some of you already like totally understand single-cell RNA-seq and like why there's a difference and what the difference is. In case you don't have that sort of understanding, I want to give you intuition for that. So imagine that we have a sample, so like this, this test tube here, which has a bunch of red dots in it that's a bunch of different cells. And this simplification, there are like 50 dots there, right? But normally a sample would be on the order of like maybe 1 million cells, maybe even more. And so we do a bulk RNA-seq experiment. And as we saw, the last 2 weeks, what you get out is a measurement of the expression of different genes. So this is just, again, like a toy example. Here we're just looking at 2 different genes, and we're looking at expression level for those 2 genes. And so here you see like the 2 genes have basically the same expression level. So what you may not have thought about before is that this is really population data. We're taking the whole collection of cells there, and we're summarizing it with, for each gene, 1 number. And so one way to think about this is, if you think about a bucket of apples, and if we took these apples, and there are lots of different measurements we could do on the apples, but simple one is just thinking of weighing. So, you go to the grocery store and you could take, a whole bag or bucket or whatever of apples, and you can weigh it. And like if you went to the checkout counter, you could like take each apple and you could hand it one at a time to the checkout person. Or like if you're doing self-checkout, you can just do it yourself. And weigh each one, and you'd get like the weight of each apple. If like the checkout person didn't first, hit you over the head for driving them crazy. And so this is what that data would look like, right? Here, it's a bar graph with each bar representing a different apple. So on the x-axis, it's just the index, it's the individual apples. The y-axis is the weight of the apples, right? And you see that you got this kind of like distribution of weights. So like I said, you could do that, and you might make it out of the grocery store without having some apples thrown at your head. But normally what we do is we go in the grocery store and we take our bucket or a bag or whatever of apples, and you put it all on the scale, and you get a weight, right? And so this weight it's a summary of all the apples that are there, right? And so here it's the average of the apples. If you took the average of the apples and paid for that, the grocery store wouldn't be very happy with you. They're going to want you to pay for all of them. But like the average of the weight is just the weight of all the apples divided by the number of apples, right? So kind of basically the same idea. And we're not doing a mathematical average, right? We're not like actually measuring all of these things and adding them up and dividing by the number. We're actually doing a physical averaging by taking all the apples and putting them on the scale. And then if we want to calculate the average instead of the total weight, then we divide by the total, right? That makes sense, right? We're getting one number to represent a whole population. And so again, at the grocery store, that's like convenient. And for a lot of things, that's convenient. But you need to keep in mind that summary statistics can be both very useful and can be very misleading. So probably a lot of you have seen before this figure known as Anscombe's quartet. And this was an observation by a statistician in the, I think the 60s or 70s named Anscombe. And he was a proponent of there was thinking at the time that, like, you know, if we have sophisticated enough statistics and, you know, computers to do statistical analysis, we can just look at summary statistics, right? We can means, medians, you know, standard you know, errors, things like that. And like, we don't need to look at individual data. And what Anscombe wanted to point out is that, so each of these plots shows different populations. And the funny feature of this quartet is that these all have the same summary statistics. So they all have the same means, the same variance. I believe they have the same medians. And so you can look at this and you can say, like, okay, well, those are very different data sets. But if you don't look at the individual data points, then you just look at the summary statistics, they look identical. And so doing this, doing bulk RNA-seq or any sort of bulk analysis on a population, whether it's apples, whoops, that was a mistake, whether it's apples or cells, there's information you're missing. And sometimes it's okay to like miss that detail. And sometimes there's a lot of important information that you're missing out. Because here, again, we're not doing a mathematical averaging, we're doing an actual physical averaging, right? We're taking all the cells and we're essentially, you know, we're not exactly, you know, we're measuring how much RNA there is of each gene. We're not directly counting the cells, but usually you'll when you do these experiments, you'll kind of have some idea of how much RNA went into the experiment, and you can kind of figure out, you know, roughly what percentage of the RNA for gene A, you know, what percentage of the total amount of RNA or the total mRNA gene A represents, and what percentage of the total amount of mRNA gene B represents. Right, so we're doing this physical averaging. And so looking at this sample, if we look at it for a single cell analysis, with these summary statistics here, again, this is basically just a mean. It could be that if we look at the data, if we measure expression in each cell, it could look like this, right? And this is kind of what this figure evokes, at least for me, is that, you know, you have expression of gene A of 50, expression of gene B of 50. And so, you know, you have this population of cells, probably all about the same, right? So this is all the cells in that tube, like looking at their individual single cell level expression of gene A and gene B. Of course, you know, for bulk RNA-seq, you'd, you know, you're usually doing all the cells in genomes, or sorry, all the genes in the genomes. You're talking about 20, 40,000 genes, but you get the idea. But you could get this data from bulk RNA-seq with the expression level of gene A and gene B at 50, and the single cell expression levels could look more like this, right? This is kind of like Anscombe's quartet. This population of cells with this combination of, you know, here each dot represents the cell again. So, right, you have some cells with an expression of gene A that's close to 100, and the expression of gene B is on the order of 40, right? Or here, I'm pointing out, you know, this one cell is coming from here where the expression of gene B is over 100 and the expression of gene A is about 30, right? So this distribution of gene expression is consistent with this bulk RNA-seq, but obviously this looks a lot different than this, right? And this is actually this sort of like difference, the the possibility that at a single cell level, gene expression might be might have much higher variance than you might capture from a population level measure like bulk gene expression has actually been understood for a while. This is a paper that, for me, actually, I think pointed out this issue, and this is back over 20 years ago, where here we're looking at gene A and gene B in, I can't remember what kind of bacteria it is. Not important, but so these are bacterial cells, and of course it made the cover of Science, so you know it's important. And what you see here is the cells are all genetically identical. They are all expressing red fluorescent protein and green fluorescent protein. And the color here is the combination of the two. So cells that are like this, where you're seeing a lot of gene A and a lot of gene B, or actually, well, yeah, if it looks like this, a lot of gene A and a lot of gene B, then it's going to be this bright yellow here. If it's expressing a lot of gene A like this, but very low gene B, then it looks bright green. If it's expressing a lot of gene B and not a lot of gene A, then it's kind of a bright red. And then like the sort of different shades in there, like this sort of darker green would be something more along the lines of this cell that's, you know, very little gene B, but moderate levels of gene A, right? So you can see this here in a sort of more visual way. And of course, like I said, this is pointed out over 20 years ago, and it's taken a while for an appreciation that that something like this does not mean that the population looks like this. So briefly going back to our apples, and here, like I said, all of these cells are genetically identical. Of course, if you're taking a sample from a human being, all the cells are essentially genetically identical, but they're not all necessarily red apples, right? You can get a sample, like whether it's a tissue sample or a blood sample, where like some of the apples are green apples and some of the apples are red apples, right? So like if you have a blood sample, that's going to have neutrophils in it, it's going to have some macrophages in it, it's going to have, you know, some circulating T cells and B cells. So those are different cell types. They're all genetically identical, but they have pretty different gene expression patterns, right? And when you take this and you do this to it, you're taking a summary of that population. And even more so if it's, you know, maybe a more extreme example where it's not just red and green apples, but you're talking about apples and oranges and pears and I don't know what else is in there, maybe some peaches, right? So like, for example, a tumor where you have like tumor cells, immune cells, you know, other non-cancerous cells that are in the tumor, it can get really messy. So for example, you could have data that looks like this. This is the same as before, I just stretched out a little bit, right? So gene A and gene B are expressed at 50, but here you have two different cell populations. And so cell population 1 is expressing gene A at a really high level, but not expressing gene B at all. And cell population 2 is expressing gene B at a really high level and not expressing gene A at all, or like, you know, very low levels, right? So this is going to look the same by bulk RNA-seq as this and as this. And just, I think, to beat you over the head with this, just one more way to think about this is that you could have an experiment where you have a control and maybe two different treatments, And again, we're looking at bulk gene expression. And here we're just looking at gene A, keep things simple. And so you can see like treatment A and treatment B both increase the level of expression of gene A, about double the level over control. And so you think, okay, well, it looks like treatment A and treatment B might be having roughly the same effect. But when you look at the single cell level, what you might see is that treatment A just increases the population, it lifts all boats, right? It increases the expression of gene A across all the cells about the same, whereas treatment B might just boost the expression in a small set of cells. And this could be like a different population, like maybe this is T cells and these are all, I don't know, macrophages or something. And so these are obviously like different, look like it's different biology. But if you are just looking at bulk RNA-seq, then you're going to miss this difference. So hopefully I've kind of convinced you that there's some value in doing single cell RNA-seq. Later on, I'll try and convince you that, you shouldn't waste your time. Now that we sort of have a little bit of the rationale for bothering to do single cell RNA-seq, let's talk about how you actually do it. I'm not going to try and compare myself with a famous Supreme Court justice, but I'll paraphrase, which is ultimately, 2 weeks ago, and especially 3 weeks ago, we talked about multiplexing samples so that you can sequence multiple samples on one sequencing lane. And a lot of the answer for a lot of problems with high throughput sequencing is just multiplexing and more multiplexing. And so essentially the way that you do single cell RNA-seq is just with a bunch of barcodes. And so instead of having sample level barcodes, well, not instead of, in addition to having sample level barcodes, we're gonna have cell level barcodes. So just as a little refresher from what I talked about 3 weeks ago, 4 weeks ago, whatever it was, this is the kind of the structure of what is, one fragment from a sequencing library. The black here is the biological sequence of interest. And then on each end, we have adapters attached. And in this case, we have an index barcode, a single index barcode that's stuck in the adapter that's on the right side of the construct. And then as part of the sequencing, we're going to get the sequence of the index barcode along with the biological sequence. So then we can go back and assign that to a sample, right? And so this is our barcode table. So we know that control sample 1 has this index barcode of the AA, GA, CC, GT, so that when we have the barcode sequence along with the read 1 and read 2 sequence, we can say, okay, well, this one came from sample 1 or control sample 1, right? And if you remember, the way that we did that was when we had the sample separate, we had separate adapters where the adapters are the same across all the samples except for in that barcode region. And so we use barcode 1 adapter, put that in the sample A tube, barcode 2 adapter, and put that in the sample B tube, so that at the back end, we can get the unique barcodes. Does that sounds, rings a bell? No need to review. Okay. So again, this is bulk high throughput sequencing. For single cell high throughput sequencing, we have this problem that instead of just barcoding samples, we need to barcode cells. So how do you do that? Well, it ends up being, relatively simple. You need more barcodes and you need more test tubes, right? So more barcodes actually isn't that hard, right? If you make the barcode longer, make the barcode long enough, you could, have one barcode for every atom in the universe. The more test tubes is slightly more complicated, but it ends up not being like super complicated. The way this is done almost always is with microfluidics. So here you have a few different things going on. So ultimately your goal is to get these micro droplets with one cell in the micro droplet, and then this bead basically contains the sequencing adapters, including each bead has a unique barcode. We'll get back to that in a second, but just to kind of show you how this works. So this is what we're aiming for. One input is the cells, and they're being flowed from bottom to top like this. Another input is the buffer that has the reagents in it. All the reagents in it except for the oligos, the sequencing adapters that are attached to the bead, and those are being flowed from left to right. And then you have oil that's being flowed in this channel, and it's being flowed from both directions, both from top to bottom and from bottom to top, so that here, at this intersection here, the cells meet the beads and the buffer, and then here they meet the oil. And so over here on the right, where you see the blue droplets, that's an aqueous droplet, and then the white here is oil, right? So the oil is essentially forming the test tube, or each droplet is its own test tube. Does that make sense? Questions about that? Okay, and so this is for 10x. This is like the device that goes on the machine that actually makes the libraries. And so each of these columns is going to represent one sample. And then I think I've never run one of these, and I forgot to double check, but I think in one row, like here is where you're going to put in your sample. Here's where you're going to put in your beads, and here's going to be where you put in your oil, or maybe it's slightly different order. But then the machine is going to, run it through microfluidics that you can't see. And then at the end of running that, then right here is where you're going to get your barcoded library out. Okay, so let's get into like a little bit more detail. So here's the beads, and I touched on this before. So each bead here, on its surface, if we zoom in, it's covered in oligonucleotides that represent the sequencing adapter. And then if we zoom in on these hairs, this is what the structure of those sequencing adapters look like. This R1 in black here, that's if you remember, for Illumina sequencing, there's the two different sequencing adapters on the different sides of the biological fragment Illumina sequence. This R1 is the like the left side sequencing adapter. And so it has both in here, it's got the priming site for the sequencing primers and the priming site for the bridge amplification, and for hybridizing to the flow cell. So then next is the 10x barcode, or what 10x calls the 10x barcode. So this is the cell level barcode. So each cell is going to get a unique barcode. We'll talk a little bit more about that in a second. Then next is what's called the UMI, and UMIs aren't unique to single cell RNA sequencing at all, or single cell sequencing at all. UMI is short for unique molecular identifier. And this is another barcode that's like, that's essentially random. And so, actually, I'm gonna come back to that in a second. And then here is the part of the oligo that's actually responsible for it's getting ligated on, or it's being used to prime off the RNA. And whether, so here it says switch oligo, it depends on whether exactly how there are different types of kits, different protocols for doing, at least for 10x, for doing single cell RNA-seq. And so this differs, what's here differs between the different protocols, but you know, it's basically the, you know, what's responsible for interacting directly with the RNA, and involved in like the reverse transcription and the amplification. So what I wanted to point out is that on a given bead, this, the R1 part, well, across all the beads, the R1 part and the switch oligo are identical. Each bead is going to have a unique 10x barcode. And then each oligo within a given bead, it's going to have random, like every one of these oligonucleotides is going to have a different UMI. Okay, so within a given bead, the cell barcode is identical, but the UMI is, you know, there's at least millions of difference of UMIs on each bead. And so like here are a little bit of details. So the 10x barcode, or the cell barcode, is about 16 base pairs long. And so theoretically, you can have 4 to the 16th different cell barcodes, right? So that's 4 billion different barcodes. And then the UMIs are about 10 base pairs long, which gives you about 1 million different barcodes. Now, this is, you might think like, oh, well, that's only 1 million different barcodes. That's actually not that many. But what you have to keep in mind is that the UMIs themselves aren't responsible for uniquely identifying. So I should back up and explain what the UMIs are doing, actually. So the UMIs are going to allow you to uniquely identify an RNA molecule from a given cell, right? So you might see 1,000 different RNA transcripts from one gene. Like, I don't know, trying to think of what my favorite human gene is. It's a tough call. I don't know, glucose 6-phosphate dehydrogenase. So you might see 1,000 different transcripts from glucose 6-phosphate dehydrogenase in a given cell. The UMI lets you uniquely distinguish, okay, well, I've sequenced this and I know it's from gene A, glucose 6-phosphate dehydrogenase. Is this actually from a different RNA molecule? I see this 1,000 times. Is each of those from a different RNA molecule, or I'm actually seeing like PCR replicates, right? So in the library prep, we're doing PCR. Did it just get amplified during the library prep? Because if it's unique from unique RNA molecules, I want to count those separately. If it's from the same RNA molecule that just got amplified in PCR, I only want to count each RNA molecule once, right? So if I see 1,000 reads of glucose 6-phosphate dehydrogenase and they're all from the same original RNA molecule, then that's going to count as one. If like they came from 3 different RNA molecules, and I'm seeing about 300 of each of them, then I can count that as 3. And if it's from 1,000 different RNA molecules, I'm going to count that as 1,000. And so the UMI allows you to look and see, okay, I know that this read is from glucose 6-phosphate dehydrogenase. I know it's from cell 3024. And are those glucose 6-phosphate dehydrogenases, all those reads from different RNA molecule are the same one? And I can look at the UMI and say, okay, well, I'm seeing these 3 different UMIs across the 1,000 reads from this gene. And so I know that I'm just going to group those into 3 different RNA molecules. That makes sense? And so that's why you have a random set of UMIs on a given bead. But of course, you need all the same cell barcode on a bead because you want all the RNAs from a given cell to be able to count back to that cell. Barcodes are unique for a cell. Yeah. So, if you see barcode, 3024, and you see you might see 10,000 different RNAs from barcode 3024. I mean, it's going to be, A's, T's, C's, and G's, right? But then you're going to, just like we demultiplex to samples before, we're going to demultiplex to cells and to some extent to UMIs. And so the reason why you actually don't necessarily need that many UMIs is because it's the combination of cell barcode, UMI, and gene, right? So if you see 2 different identical UMIs, but they're attached to, like one's attached to glucose 6-phosphate, a read from glucose 6-phosphate dehydrogenase, and another one's attached to a read from, I don't know, the insulin gene, like that those can't be the same because they're different genes. They came from different places in the genome. It just happens that, we have a relatively small set of, 1 million different UMIs. And so you might have the same UMI associated with different genes within a given cell, but it's unlikely that you're going to have those 2 different UMIs associated with 2 different RNA molecules from the same gene in a given cell.
So what part of the oligo binds to the RNA?
It's the switch oligo.
Okay. Those are going to be kind of designed for given genes, or
No, it depends on, so the easiest way I can explain what the switch oligo is. The easiest way to think of it is that for one way of doing the initial, so the first step, what actually happens on this cassette is actually making a cDNA library. There's a separate step of making a sequencing library. And so what happens there, you can either start from the 3' end of the RNA or the 5' end. Usually, if you just want to look at sort of basic gene expression, we'll use from the 3' end. And in that case, you use this oligo here is a poly DT. So you're basically grabbing onto the polyadenylation on the end of the RNA. The switch oligo, if you want me to, I can explain it, kind of what I remember. It's a little bit more of a trick, but it's a similar idea. So it's totally not gene specific. But it's for the switch oligo. I just happen to choose this one. That somewhere else there's another figure that shows the poly DT. But yeah, it's the switch oligo is for the 5' approach, which for some more complicated assays, like if you want to do, I'm going to talk about it in a second. Like if you want to do like T cell receptor sequencing, you actually have to use the 5' approach. Other questions? I feel like there's something else I was going to mention. Yeah, maybe we'll stop there for now and take like a 5-minute break. Well, while we're stopping, if you want, you can ponder, how do you actually get a bead where every oligo on the bead has the same cell barcode? but each of these beads should have a different cell barcode. Okay, let's take 10 minutes. So I have like 2:03. So let's say we'll come back at 2:13. So just to give you a little bit of an idea of what you can do with single cell, all these are RNA-seq, yeah. So single cell RNA-seq. So I think when most people are thinking about single cell RNA-seq, they're thinking of gene expression. So the same sort of approach that you would do with bulk RNA-seq, but you're getting it at the cell level. You're getting the gene expression details at the cell level. And you can both do like sort of just looking at differential expression, but also part of what people are often doing is trying to, within the population that they're sequencing, trying to cluster cells into cell types, right? You know, so if you had like, just like a blood sample, then you would, you know, you might try to separate out the different types of cells that you'd expect to find in circulating blood, right? So like T cells, B cells, macrophages, things like that, and cluster them. And then you could, to some extent, look at those clusters independently. You might even have subclusters of like different subtypes of T cells. And so that is, I think, the most common application of single cell RNA-seq, but there are other things that you can do. For example, and often people will do the gene expression in combination with these other, one or more of these other applications. So you can do a TAC-seq to look at basically epigenetics, to look at the chromatin accessibility. So like parts of the genome in a given cell that are actively being transcribed as opposed to, or that are accessible for transcription versus being kind of locked down. You can do cell surface proteins, which is basically gives you sort of similar information to what you get from flow cytometry. But of course, if you combine that with a TAC-seq or gene expression, then you get cell surface proteins. So you can like divide cells by their cell surface proteins and then look at the gene expression within populations based on the cell surface protein. You can do sequencing of, in B cells, do B cell receptor sequencing, or in T cells, you can do T cell receptor sequencing. You can look at the specificity of T cells, although that's, it's not like, you basically have to like design specific, you know, like you can identify T cells that have T cell receptors specific for a given, you know, given peptide, not like you get everything. And you can do spatial gene expression where instead of, you know, what we talked about so far and like what you know, normally doing for bulk RNA-seq is you have some cells in suspension that you're looking at their expression. But with spatial gene expression, you actually would have a microscope slide, you know, thin section of microscope slide, and different regions within that microscope slide, you can get expression. It's a little bit more complicated, really, for doing like the single cell RNA-seq approach. You can't get individual cells. You usually get like sort of these clumps of 2 to 10 cells. There's a newer platforms that 10x and another company have come out with that do allow you to get subcellular, but it's a targeted, you know, looking at certain specific genes that you decide on, not like a sort of shotgun approach like you would with standard bulk RNA-seq or single cell RNA-seq. And I don't know if I've mentioned this before or mentioned this already. I think I glossed over it. So I keep on referring to 10x. So there's a company, 10x Genomics, that's the biggest maker of equipment and reagents for single cell RNA-seq. There are other other companies, I think other companies still in business, and also like, you know, different research labs have developed methods that you don't have to use the 10x platform. It's just, you know, the most popular. So that's, you know, most of the single cell RNA-seq data out there is coming off 10x machines. So just to give you an idea of how these other, some of these other, like the T-cell receptor sequencing, the cell surface proteins, B-cell receptor sequencing works. So that on the left here, it's this is the cell surface protein approach. I was lazy and stole this figure from, or both these figures from, like 10x literature. But I will point out that whoever made this figure doesn't actually know how antibodies work, because the little pointy end should be bound to the, this green blob and the yellow blob and the blue blob are cell surface proteins. And the pointy end should be attached to the protein, not like on top of it. But I understand that, obviously the graphic designer didn't. So the way, and so this is cell surface proteins. Here is that looking at the T-cell receptor specificity. And so they both work in a similar manner in that for the cell surface proteins, I told you it's kind of like flow cytometry. It's very much like flow cytometry because you have antibodies that are specific for different cell surface proteins. And each of those, let's say here it's 3 different antibodies, right? So let's say, you know, the yellow antibody is specific for the yellow protein, the green antibody is specific for the green protein, and the blue antibody is specific for the blue protein. And so on the blue antibodies, you would label those with a oligonucleotide tag that's like the blue specific tag. And the green antibodies are going to get the green specific tag, and the yellow antibodies are going to get the yellow specific tag. And so you actually do this labeling before you do the microfluidic step that I mentioned before. And then when the cell gets in that droplet, that I guess I glossed over this. Do I have other slides? No, maybe I didn't. I should have given you a little bit more detail. So once those droplets are formed, the cells get lysed within the droplets. So, you know, all of the RNA within the cell gets spit out into this droplet. It's essentially this little test tube. And then the reagents are in there to make the cDNA library. So these antibodies with their oligonucleotides are in that droplet. And so the part of the library it gets, you know, the yellow antibody. If you had yellow protein on the surface of the cell, you'll have yellow antibody bound to the surface of the cell. You know, it's quantitative, just like for the RNA. And so if you have like, you know, 10,000 of the yellow antibody oligonucleotides, that tells you have, you know, roughly 10,000 yellow antibodies on that cell, which tells you there's, you know, roughly 10,000 yellow proteins on the surface of the cell. So it's kind of a cute thing that you know, you just stick in some DNA there and you figure it out. And similar for the T-cell receptor specificity is you have these, because the interaction between the T-cell receptor and the peptide is fairly weak, you don't have like unlike the antibodies, you don't have like a single antibody with a barcode. You actually have these like conjugates of multiple peptides on this, you know, some sort of flexible linker. And so you need multiple of these peptides to actually bind to the surface for it to stick. But then again, you have this on this like complex of peptides, and this complex of peptides, you also have an oligonucleotide that uniquely identifies it. So you could have, I don't know how many, I haven't worked with this sort of data, so I don't know how many different like peptides you can test, but just like you'd have different oligonucleotides on the different antibodies here, you'd have your different peptides with different specificities, and you'd have a different DNA fragment there that would label what the peptide specificity of the T cell receptors on the surface of that cell are. Does that make sense? And so downstream, after you make the cDNA library, which is what happens on the machine, then for each of these, for each of these, you actually end up making a separate sequencing library. They're all linked together because for a given cell barcode, that's going to show up in your gene expression library. It's going to show up, that cell barcode is going to show up in your T cell receptor library. If that's what you did, it's going to show up in your cell surface protein library. So you can link all that back to a given cell. Questions about that? Okay. So I just told you how fantastic single-cell RNA-seq and related single-cell technologies are. Like, you're all sold, right? You're all going to go out and do single-cell RNA-seq. So let me tell you why you shouldn't. And I don't get paid by the company. I can complain, about 10x all day. So it does give you a lot of useful information at the single cell level as opposed to the bulk level. And so you understand the population a lot more. It is, however, extremely expensive. I haven't looked at quotes recently from the Duke Core. It's been probably a year or two since I did. The cheapest that you get from them, I think it's just for the library prep. I don't think it even includes the sequencing, is about like $2,000 per sample. Whereas for, I just for a project sent in 35, 40 samples for RNA-seq, just bulk RNA-seq. And that project, I think for all of the RNA library prep and the sequencing is like $5,000 for 40 samples. Whereas for single cell, if I was doing single cell on those samples, you'd be talking about like whatever, $80,000. Now, and that $2,000 per sample, it depends on, that's the sort of, you get a multi-sample discount, right? So if you have 8, and the numbers might have changed since last time I looked at this, but it's a lot more expensive to do single cell. It is noisy for at least 2 reasons. There's biological noise, because it turns out that, well, that's kind of the point, right? I mean, I showed you this figure that like, even in this population of like clonal cells that are in the same environment, like right next to each other, these aren't different cell types. You get differences in gene expression. So you can imagine that you have this population, you take cells from blood, even within the same cell type, you get noise. And so whereas, there's a concept of, say, housekeeping genes, or like, to go back to the example of say, T cell receptors, you think, okay, well, it's a T cell, it's a mature T cell, it has a T cell receptor, so I should be able to see like the T cell receptor gene. Now, you actually can't always see the T cell receptor gene in a given T cell, because because the expression of proteins can be noisy, and 2, because for some proteins in a given cell that that protein is there, the proteins might be very stable, and so like at a given point in time, the cell might not be expressing the RNA for that protein. So you get noise, you get that biological noise. You also get technical noise because you're taking, say on the 10x platform, you're taking about 10,000 cells and you're barcoding them individually, and you're trying to, sequence the RNA, and you just go between the, the amount of sequencing that you're going to do per sample and just sort of like technical variation. You're not necessarily going to capture all of the RNA, or you're almost certainly not going to capture all of that RNA on the cell in the sequence. And so you have, like I said, you have this biological variation, but you also have this technical, or you have this biological noise, you also have this technical noise. So you end up seeing a lot of what's called dropout in single-cell RNA-seq, where you see genes that, for a given cell, you might not see any expression for a gene. And of course, 20,000 genes in the genome, you definitely don't express, if your sample is a blood sample, you don't necessarily expect, or you don't expect to see expression of neuron-specific genes in your blood sample. Okay, but again, you also don't see expression of, T cell specific genes in some of the T cells. And disentangling that like biological noise from the technical noise is, there are approaches for doing that, but it's not great. And the flip side of the, what I pointed out here, which is that, well, I'll point out here several times, which is that you're doing experimental averaging, so that this and this looks like this. That's a problem if there's a lot of variation in the sample. But biological averaging, or like experimental averaging, you're getting an average. So you actually you're averaging out a lot of the noise. So that's the plus side of bulk RNA-seq, is that you're experimentally averaging, so it ends up being less noisy. But that experimental averaging comes at a cost of understanding. So expensive, noisy. You're working with small populations. So one sample for 10x, pretty much the most that you can get from that sample is about 10,000 cells. And that's like the upper limit. You don't always get that. And so, 10,000 cells ends up maybe not necessarily being that many cells. So you're ultimately working with small populations. I mentioned, kind of at the beginning, for bulk RNA-seq, often your samples are 1 million cells. So you get this averaging, but you get this averaging over more cells. And so not only do you get the, you're getting this sort of experimental averaging that helps with the noise, but you're averaging over a larger population. The sample requirements for single-cell RNA-seq can be kind of tricky. This has gotten a little bit better, but depending on what you're doing, you really actually have the sort of, for the standard 10x prep, ideally you want fresh cells that ideally haven't been frozen and thawed, which means that you're collecting your cells and then you're taking it to the machine right away, within a couple of hours. That can be really tricky. They have protocols that kind of get around that. So instead of doing single cell, you can do single nuclei, which has pluses and minuses, but it's tricky. If you're doing single cell, you have to be able to get those cells disaggregated. So if it's a tissue, you need a way to break up the tissue without breaking, lysing the cells, which is tricky, unless you want to do spatial single cell. So that's problematic. So it's less forgiving. Single cell RNA-seq is definitely less forgiving on samples than bulk. And then for the most part, you don't really get splicing information because, actually, this is another, I'm going to get to it in a second. The part of the RNA that you're sequencing is usually a pretty small bit. And for this, the basic protocols I mentioned before, where you're doing 3' sequencing, it's always at the 3' end of the gene, right next to the polyadenylation. And so unless the splicing is right there within the 70 base pairs of the polyadenylation, you're not going to see the splicing. There actually is, I talked a few weeks ago about different high throughput sequencing platforms other than Illumina. So there are now protocols for taking 10x libraries and sequencing them on, I know you can sequence them on the nanopore. I can't remember if PacBio also has it, but you can actually do long read single cell RNA-seq. I've never worked with any of that data. I think it's pretty niche now. I assume it's super expensive because already the single cell is expensive. But it is essentially, it is becoming feasible. So, which is why I put an asterisk there. Questions? Okay. So that's kind of the overview of you know, how single cell RNA-seq differs from bulk, some advantages and disadvantages. So I'm going to give you the like sort of very high level overview of how we analyze the data differently than bulk, at least at the bioinformatic level. And then, like I told you, for the next 2 weeks, Tyler is going to then walk you through the sort of the more exploratory data analysis and you know, statistical analyses that you can do once you've done the sort of initial bioinformatic analysis. Okay, so this should look familiar to you from 2 weeks ago. When you have bulk RNA-seq data, you're starting off with sequence data, and for the bioinformatic analysis, the goal is to get to this, where you have a table for each sample. You have the counts for each of the genes in the genome, right? So like here, sample 1 for gene 1, it has 0 counts. For gene 2, it has 5 counts. For gene M, it has 0 counts. Get the idea, right? For single cell RNA-seq, it's really the same goal. It's just that you can think of it as adding another dimension or just sort of like stretching this sample dimension so that here, you know, you have like for each gene, you have one count value for each sample. Whereas here, for each sample, instead of like taking the sample as a whole, you're subdividing that sample into individual cells, so that for sample 1, cell 1, you have 0 counts of gene 1, 5 counts of gene 2, 0 counts of gene M, right? And then for cell 2 in sample 1, you have 9 counts of gene 1, 0 counts of gene 2, 0 counts of gene M, right? So in some ways, the data is pretty much the same. It's just that, like I said, instead of having, you know, 1 test tube per sample, now you essentially have like 1 test tube per cell, and you can then group those cells into samples if you want. I mean, usually you do want to, right? But your sort of unit of measure becomes the cell instead of the sample. And so, again, a couple of weeks ago, we walked through how you do the bioinformatic analysis of the bulk RNA-seq, and I kind of broke it down into this sort of pre-processing and QC, the mapping and counting step, the mapping step where you look and see for each read where in the genome it came from, and then you like add for each read that maps to a given gene, you add a count to that gene for the sample. And so for single cell, it's pretty much the same thing. You just add in these additional steps where for each read, you you map it, then you assign it to, actually, I don't know if I have the order right, I just plugged it in there. But you map the read to a gene in the genome, but you also assign the read to a given cell. And within that cell, you assign it to a given RNA molecule, right? Because remember, we have the UMIs that we can trace back a given read to an original RNA molecule that was in the cell. And so then, of course, you, like I said before, if you have multiple reads from a given RNA molecule, then ultimately you're going to collapse those down. You can do that. I hadn't mentioned this before, but you can actually do that for bulk RNA-seq also. You can assign UMIs to RNA molecules. Sometimes people do it, often they don't. It's a little bit, technically it's not that complicated. You just have different adapters. Usually those kits are a little bit more expensive, and you are paying to sequence the UMIs, but it's similar idea. So this is like breaking down those different steps for bulk RNA-seq. I guess I should have said that as part of comparing bulk to RNA to single cell. That's probably obvious. So the first step, that sort of QC pre-processing step here, the software is FastQC, MultiQC, and Trimmatic, or at least that's what we used. And then the mapping and counting are both done by STAR. And you already seen this, like the output of that first pre-processing step is a FastQ, the output of the mapping step is a BAM file, and then the output of the counting step is CSV. For single cell RNA-seq, I'm simplifying things a little bit, but the sort of default way of handling this is using a pipeline called Cell Ranger that was developed and made available by 10x, the company that makes the machines and the reagents. And so basically everything gets done by this function Cell Ranger count. So you give it the FASTQ files, and it does some QC, it does mapping, it does counting, it does that cell barcode assignment, and that UMI, RNA molecule assignment. If you, depending on how you get your data, you might have to do this first step of Cell Ranger MK-FASTQ, which does demultiplexing, and sometimes you'll get data, instead of getting it as a FASTQ, you'll get it as a BCL, which is BCL is the sort of native proprietary format on the Illumina machines. So most of the time when you get data from Illumina sequencers, you get it as a FASTQ, but sometimes core facilities will give you a BCL for single cell data, and then you need to, they're it really doesn't matter too much, but sometimes they'll give you the BCL for single cell data, and then you need to convert it to a FASTQ as well as demultiplexing. Sometimes you'll get the FASTQs already demultiplexed, at least by sample. This is one of the few cases where these days there's a decent chance you'll get multiplex data returned to you by a core facility. But both of these are essentially black boxes, which I find somewhat annoying. There are other pipelines that have been produced by academic labs for analyzing single cell data. They have their pluses and minuses. In my experience, if you just want to do sort of gene expression, basic single cell RNA-seq, then those other pipelines work pretty well. If you want to do any of the fancier stuff like the T-cell receptor sequencing or the B-cell receptor sequencing, like the cell surface proteins, you kind of have to, as far as I know, or at least last time I checked, you kind of have to go through the 10x pipelines. And of course, 10x does update the pipelines semi-regularly. They're, like I said, they're kind of black boxes. They tell you kind of what's going on inside, but not really. You don't have that much control of what's happening. And the software is kind of slow, but it is relatively easy to do the bioinformatics with Cell Ranger. And so that's actually what we're going to do in a little bit when we get there. Okay, so I want to show you what like actually fragment that's getting sequenced for single cell looks like. But just to remind you, you've already seen this figure like 10 million times at this point, right? This is this black part in here is the biological sequence. It's actually like the sequence of interest, and this is sort of bulk RNA-seq, standard DNA-seq, stuff like that. Right here's the P5 here is, and the P7 here are the oligos that are used for the bridge amplification and hybridization to the flow cell. Here's the sequencing primer, or the sites for the priming sites for the sequencing primers are in red and purple. Here's the barcode index. So keep that in your mind. This might have probably did on the same slide, but I didn't. So here's what the structure that you're going to see for a fragment for 10x single cell RNA-seq. So colors are different, but here you have the P5 and P7, right, for bridge amplification and hybridization of flow cell. What's labeled here is read 1 and read 2. Those are the priming sites for the sequencing primers. So it's here what's in red and purple. And then, so you can also have, and often you'll have per sample index barcodes, right? And those are just like these. Then the differences are, well, this part is actually the same. This is the biological sequence, which is the same as here. The size of the insert here is usually pretty small. And then the other stuff here is what's specific to 10x. So here's the 10x barcode. That's the cell barcode. Here's the UMI, and then here's the whatever bit was used to capture the RNA. So like if you're doing 3' sequencing, then it's going to be the poly A sequence. If you're doing that 5' sequencing, it's going to be the part that's specific to the switch oligo. And this actually you don't really care about. It's kind of garbage sequence. It's technical sequence that you needed to actually like construct the library, but it's not interesting. And so often the data that you get from 10x sequencing, the R1 read is sometimes if the sequencing facility is flexible, you can just, you would say to them, okay, I just want 26 base pair reads on this. I don't need 50, 100, 150 base pair, because I just care about the first, I care about the 16 bases of the cell barcode and then the 10 bases of the UMI. Like nothing else is interesting here. And then read 2 is the biological sequence. And then again, those will often be short, like 100 base pair, 75 base pair. And so like you can like save some money. Instead of doing 150 base pair paired in reads, you can do 26 forward reads and 75 100 base pair reverse reads. Does that make sense? Yep.
So if you're doing reads from both directions here, what's like the technology doing the sequencing? Does this feel like, because it's a bead, so it's not the next gen sequencing.
No, it's next-gen sequencing. So by the time you get here, it's just, as far as the sequencer is concerned, it's just like any other sequencing library.
Oh, flow cell or no?
Yep. Usually it's on Illumina. Like I mentioned, there's like kits and protocols for doing this on Nanopore, but like the vast majority of at least 10x data and probably single cell data in general is just Illumina, right? The structure of this is a little different in terms of how you decode it, but as far as the sequencer is concerned, it's just paired end, and you don't have to, you know, I said you can do 26 base pairs here. You don't have to, you can just give it to the sequencing facility and say, like, give me 100 base pair paired and read. Actually, the data set that we're gonna look at a little bit today, we'll do the pre-processing today, and that Tyler's gonna dig into more next week. I noticed that they did 150 base pair paired end reads, and so you actually get, like, on the R1, you have your 26 base pair of you know, cell barcodes and UMIs, and then you actually do have, like, the rest of it I blasted because I was like, what are they doing with 150 base pair paired end reads? That the DuCore doesn't give you that for 10x. They actually give you, you know, 26 and 75 or 100. I blasted it, and like the R1s, you get like kind of only the 3' ends of the reads actually map to the human genome. The 5' 30 base pairs don't, and that's because it's barcode and it's 10x barcode and UMI. But there's no reason why. When I say you can ask for 26 base pair reads, that the sequencers are programmable to that extent. Like, you can set the default is that you do, if you do paired end reads, you do equal numbers, 50, if you do 50 base pair paired end reads, it's 50 base pairs from read one, 50 base pairs from the, like, this priming site, and 50 base pairs from this priming site. But that, they actually have the flexibility that you can change that. Does that clarify? Okay. Other questions? Yeah, I don't know why for this data, I don't know why they did 150 base pair. It's possible that wherever either they didn't know that they didn't need to do it, or wherever they actually had a sequence just didn't offer that. I mean, some of the, like the big sequencing companies like Novogene, they don't use, they sell 150 base pair paired and reads. If you want 100 base pair paired and reads, here's your 150. I mean, it's like the Model T, right? You can have it in any color you want as long as it's black, right? So you can buy from them 150 base pair paired and reads, or you can buy 150 base pair paired and reads. Okay, and sorry, needing the tea. Sorry to kind of walk through this, right? But for the bioinformatic analysis, then once you have this data, this part is going to allow you to group it to cells. The red here is going to allow you to group it to RNA molecules, and then the insert here, which is the biological sequence, is going to allow you to map it to the genome location. And so what those files that are coming off the sequencer look like is, like I said, usually the R1 file will just have the cell barcode and UMI. Again, it's not going to be the case with the data that we're looking at today. But I mean, it does have the cell barcode and UMI, but also has biological sequence. The R2 is just going to have the RNA. And then if you multiplex samples, which usually you'll do, then you'll get an I1. And even actually, I think for our data today, I was looking at this recently. I think it's the data today. There's an I1 and I2, so it's actually dual indexed. Right, and so that allows you to demultiplex samples to demultiplex reads to samples. Question. Okay. So let me tell you a few details that I glossed over before. So number one, I told you that you really want to have your goal with microfluidics, and here's a slightly simplified version of it, is to have in each droplet, you want one bead and you want one cell. In reality, that's not what you get a lot of the time. So it depends on the concentration of cells and beads that you put in, and that's obviously adjustable, but it's a random process. So usually what you do is you put in beads at a concentration where most of the droplets don't have any cells, because that increases your likelihood of getting mostly droplets with one cell. Because if you have a droplet with two cells, now you're back to bulk RNA-seq. I mean, it's 2-cell bulk RNA-seq or 3-cell bulk RNA-seq, but you're still not getting single cell level information. And that's gonna, it's a random process. And so you'll get some number of droplets with one cell. You'll get a bunch of droplets. If you do it right, you'll get a bunch of droplets with 0 cells. But then you'll get some droplets with 2 cells and some droplets with 3 cells. And so this is actually, this is data, I can't remember if this is from a paper or a reference here. I think this might originally be from 10x, right? So the number of cells that you get out the back end, the more cells you load in, the more likely you are to get doublets or multiplets, droplets with multiple cells in it, right? So there's this trade-off, right? If you go for a low number of cells, right? So your data set for a given sample, you're going to say like, okay, I'm willing to take, that I'm just going to get 500 cells out the back end. Then you get, your predicted rate of multiplets. So 2 or more cells in a droplet is pretty low, like, it's less than 1%. If you want to like sort of max out the technology, so you get, as many cells as you can for your dollar, then your estimated rate of multiplets is 7%, which is actually pretty high. I mean, I think, you're only getting 10,000 cells, right? And so there is, so this is like the technical limitations of the technology. So within Cell Ranger, so independently, academic labs have developed software for like detecting multiplets. And that's actually been baked into Cell Ranger, which you can turn it off if you want to do that sort of multiplet detection on your own, you can. So it does do a pretty good job of identifying these multiplets and filtering them out, but it's not perfect. And it does mean that you're throwing out data, right? Basically, the assumption here is anytime you have a droplet that has more than one cell, it's kind of useless to you because you're now averaging over those. You don't know, if this is like one of the cells is a T cell and one of them is a macrophage, then you're averaging over 2 different cell types. So that's just there's a limitation. It's something that I think gets glossed over a lot, but it is a problem with the technology.
So there's more often going to be multiple beads in a sphere than there are multiple cells? Fewer cells.
It's a good question. So I'm not sure of the answer to that. So you'd actually not a good way to detect multiple beads.
Then you get multiple B cells. One B cell.
Yeah, it's a little, in some ways it's a little bit less of a problem because it's a little bit of a problem because you're basically having a repeated measures of that cell. But I certainly feel less concerned about that than multiple cells. For sure, you must get, I'm trying to think if I've thought of this before. For sure, you must get multiple beads per droplet. But yeah, I think it's less of a problem. I mean, it certainly, I'm less concerned about it. But yeah, good question. Should be able to, I'm trying to think if you can.
I know percentage of mitochondrial G something used for QC, but I don't think that's not QC. It's like for multiplex or.
So usually the percentage mitochondria is used. It is actually used. I think that Cell Ranger, does it filter on that? No, I guess not. Ask Tyler next week, because this is something we've been through. I think that what he normally does is actually do the mitochondrial filtering downstream, and I can't remember if Cell Ranger does anything, but the mitochondrial filtering is actually usually focused on, it's actually, the multiplet filtering is somewhat similar approach. I can talk a little bit about it. But the mitochondrial filtering is because there's an association between high mitochondrial, if you have an elevated mitochondrial gene expression relative to nuclear gene expression, then that is a sign that the cell is undergoing apoptosis. And therefore, usually, that's not interesting, because the like gene express, when a cell's undergoing apoptosis, like one of the biochemical aspects of that is the DNA and the RNA gets degraded, and so not interesting. You also get, like, if a cell is successfully undergoing apoptosis, then you get these little exosomes, basically, of like subcellular exosomes, right? That's like got captured some part of the cell, right? But it's not really a real cell anymore. Right? You might have a bunch of those, and they're not really biologically. They're not a good biological snapshot of what's going on in the cell. Yeah, it's a dying cell, and it's also like might not even like a whole cell anymore. So yeah, that's why people look for do like a sort of this basic filtering of gene expression of mitochondrially encoded genes. Right. Other questions? All right. So I just have one more slide, and then maybe we'll take a break, and we'll come back and do the hands-on stuff with Cell Ranger. We might even end early. Let's see. So the one other step that's like kind of a little detail is that you need, for Cell Ranger at least, which is what we're going to be focusing on today, and I guess Tyler might touch on it a little bit next week, is that you actually need like a specially processed genome as input to Cell Ranger. And so you have to, like, usually if you're working with human samples or mouse samples, the easiest thing is just to download the genome from 10x. They have a database of common reference genomes that have already been like specifically processed for Cell Ranger pipeline. If you're working with a like less popular organism or a non-model organism, then there is a tool that's part of the like Cell Ranger toolkit for processing a genome sequence and annotation so that it's properly indexed and packaged for Cell Ranger to use. it's like a little bit of a hassle. That's not actually that big of a deal. But so that tool is called Cell Ranger Make Graph. Okay, so it's 3:04. Let's take a 10-minute break. We'll round it off. We'll say 3:15. I am going to, because I have it here, remind you that if you haven't signed in, please sign in. If don't take the survey yet, but like you can go ahead and open the webpage so that it'll be ready at the end, and hopefully I will remember to remind you. And if you don't have Open on Demand open, then you should do that now. I'm going to set the timer and see you back at 3:15. Okay, so there's a question in the chat about why UMIs are used in microRNAs. Sorry, for microRNA bulk RNA-seq. And so if I'm understanding the question correctly, I can kind of answer that. It's actually been a long time since I worked with microRNA data, but usually there are kind of two main, two kind of big applications for UMIs. One is like in single cell RNA-seq, where you want to be able to avoid double counting what are called PCR duplicates. So where you have, there's an amplification, you know, a PCR step, or I guess it could be a linear amplification step. And you don't want to, if you have two reads that actually came from the same initial RNA, you don't want to double count those because your goal is to figure out how many like original RNA molecules there were. And so if you have two reads that look identical and they have the same UMI, then you usually will collapse those down into one. The other reason why UMIs can be used is if you're trying to like improve the accuracy of sequencing, then you can look and you can collapse reads into UMIs and like either take a consensus of those multiple reads, take the sequence consensus of those multiple reads, or you know, there are a couple of other approaches to try and like use that sort of multiple measurement information to improve the sequence quality or the sequencing accuracy. So for microRNAs, I'm guessing that those are being used for the that people are using UMIs for the multiple counting to be able to collapse down reads that came from the same multiple reads that came from the same molecule down to a single count. But I'm not 100% sure without doing a little background reading. Other questions? Okay. So I am done with the slides, I think, for now. And so let's go back to Open on Demand. And what you should be seeing is something like this, where you have a dialog box that says pop-up blocked. And if you have that, then that's actually good news. It's for whatever reason, RStudio, when you knit, the web browser seems to, it wants to show you a window that's the result of the knitting. And it seems like most web browsers view that as a pop-up. And so, which I guess it is. And so if you haven't actually configured your web browser to allow pop-ups from DCC on demand, then you get this message and you can just click on try again. And usually then it works. Of course, it's not working now. There we go. Okay, so, and this is the report that's coming from RStudio, and most of it's actually not interesting because our goal here was to run Cell Ranger, and we'll actually look at that in a second. The things that are kind of interesting, actually we can, how do I want to do this? We have this open. We'll look at this first. Okay, there's a chunk of code at the top that just actually allows each of the subsequent chunks to be timed, which was kind of useful when I was trying to figure out how long this is going to take to run. This is, we're going to look at this again in a second. Actually, no, let's look at it now because it's right there. Okay, so Cell Ranger gives us two outputs that we care about. One is the actual like matrices that we're going to use as input to the downstream analysis. We're not going to touch those today. And the other is it gives you a little report that's the sort of QC information. And that's what we're going to be looking at. And so the easiest way to get to that, actually, ignore what I just said. We'll come back to that in a second. Because otherwise, once we close this window, it's going to be a pain to find it. So this is actually the chunk. We'll look at it more carefully later, but just because we have this pop-up up here, we'll look at, we're going to go through some other stuff first. So this is actually the chunk where Cell Ranger got run. And this is like all the output that Cell Ranger gave us. I don't know why it's being so laggy here. So this is, you know, a bunch of like sort of log information as it was running. It's not very interesting. I'm gonna like actually try and scroll faster. And then like the last little bit that we want to look at is here it's telling us like how long it took to run, and it took almost 5,000 seconds. So a little bit longer than I expected, but you know, about 75, 80 minutes. So this we ran on one of the samples from the data set that you're going to be looking at next week and the week after. And I specifically picked the smallest sample so it would run the fastest. And it's actually a good bit smaller than the others. So you're not going to, we've already like sort of pre-processed all the samples. So next week we'll, you know, kind of do it like cooking show style where we'll already have like sort of baked the samples, and you'll be ready to, you know, ice the cake. You don't have to worry about baking the cake. Okay. So now if you scroll back almost all the way to the, or I guess all the way to the top, you'll see there's one line here that says report will be named web summary.html in, and it gives you a directory. So what I want you to do is I want you to select that directory. We're going to find the report. Your path will be a little bit different than mine, but it should be pretty much the same. So on a Mac, you can do Command C. After you've highlighted it on a PC, you should be able to do Control C. And then we're going to go back to the RStudio window. And you want to click on the three dots that are here. Let me make this a little bit bigger. Maybe scoot over my computer so I can see the screen. Okay, so you want to click on the three dots, and that will pop open a dialog box here where you can say, where it says go to folder. So now you can paste in that path that you copied from the report. And so on a Mac, it's going to be Command V. On a PC, it should be Control V. Anyone not got this, where they have the paste it in. Okay, great. And click OK. And then that's going to teleport your file browser here to the output directory from Cell Ranger. And so actually the stuff that you're going to be working with next week are this matrix, the filtered feature BC matrix H5, I'm actually not sure what the molecule info, I don't remember. The raw feature BC matrix, I'm trying to remember what else. Well, I don't think you end up using the BAM. Oh, the metric summary, I think, ends up getting used. But Tyler's going to talk about that next week. What we're interested in is this web summary.html. So if you click on that, then it's going to give you the option of open in editor or view in web browser. You definitely want to do view in web browser because if you open an editor, you're just going to see the text of an HTML file, which is not interesting at all. And so this is what the report looks like. There's like a kind of alert message up here that maybe we should pay attention to, but we're going to skip over. Basically, just like a change that they've made in how Cell Ranger works. And you get, so there are actually two tabs here. We'll look at this one really quick and then we'll flip over to the other one. So here, this tab, it's estimating the number of cells, right? And so that's coming from the cell barcodes, although it's a little bit more complicated than that. And I'll talk about that more in a second. It's estimating the number of reads per cell, so about 30,000 reads per cell. And remember that, well, actually, yeah. So, and then it gives you that median genes per cell, right? So it's detecting about 2,000 genes per cell with about 30,000 reads per cell. Now, remember that like a read, not only can you have, for a given cell, not only can you have multiple reads coming from a given gene, but you can actually have multiple reads coming from a given RNA molecule, right? They can have multiple reads that have the same UMI coming from the same gene. So, you know, this ends up being about 10,000 reads per cell on average. But these are not necessarily, you know, even unique transcripts, right? So some of these could be duplicates from the same RNA molecule. Here's some details that actually we'll see got put in at the beginning, the sample. There are several, I mentioned this before, there's several different kits for 10x. And so this is the 5' sequencing. That was information that we put, I think we put it at the beginning. I can't remember if we have to put it in or to text it. I think we put it in. Like the reference genome that was used, the version of Cell Ranger, total number of reads, you know, percentage of barcodes and UMIs that were valid. I think sequencing saturation has to do with like how much is an estimate based on the rate at which you're seeing the same UMI. You know, basically the rate at which you're repeatedly sampling the same RNA transcript, basically. And this is sort of like quality information that's not super interesting.
That's 60% mean that the sequencing saturation mean that 40% are first time it's encountered that, and then not even.
So I think I'd have to go back and check. I think what it means is that it's estimating that if you sequence this infinitely deeply, what you would find is that for this library, for this data set, you'd sequence 60% of the unique library fragments, right? So there's a 40% of the library that you haven't seen, right? And there's going to be diminishing returns, right? If you sequence twice as deeply, you're not going to get to 100%. I mean, you probably, right, there's a sampling issue. So I'm pretty sure that's what that means, but I'd have to go double check. I could be wrong about that. Yeah, I think it might be a Poisson distribution of sampling. And so it's sort of estimating the parameters of that curve based on the like multiple count, how many like multiple hits you get. Reads map to the genome, you know, basically it's saying that like 30% of the reads like it didn't map to the genome. That could be like sequencing error, it could be contamination, it could be a lot of things. So this is actually where this plot is where this number comes from, this estimated number of cells. So there is an algorithm that is used by Cell Ranger under the hood to, based on the relationship between UMIs and barcodes, that it uses to decide like what's unique cells. So remember I told you that you get droplets that have beads but no cells. So those droplets have beads but no cells, but they still can have some RNA in them because cells that get lysed before they're in a droplet, you know, if they spontaneously lyse during processing, that RNA is going to be released into the buffer and it'll get in a droplet. You can also get like sort of bogus. Ideally, the way the beads are designed is that if there's no RNA, if you have a droplet with no RNA in it and just have a bead, you shouldn't get any signal from it. But of course, it's, you know, it's biology and chemistry, and so it's not perfect. But if you have like contaminating RNA from lysed cells, then you're going to get a signal from that. And so it uses the number of unique UMIs and the like kind of number of genes that are detected within a droplet to try and estimate basically where to draw the cutoff for UMIs per cell barcode, at which it says, okay, well, you know, if I'm only seeing 10 UMIs, 10 unique UMIs in a given cell, then that's probably not a real cell. That's basically an empty droplet. And so here is this is where you see it transitioning from like black, although it looks like partially blue, I don't know why, but where you see it transitioning from black to gray, that's where it's decided to draw the threshold. And usually it's like this, right? It kind of goes, you know, there's sort of this gradual distribution from like a high number of UMIs per cell to a relatively low number of UMIs. And then like where it starts dropping off, it actually always seems to me that it's like a good bit after it's dropped off. I don't know exactly how, what the algorithm is, but that's where it's deciding the cutoff, right? So basically everything, all the cell barcodes here and above are considered from real cells. So there are 2,300 or something of those. And then like below here is just garbage. And I will say that like the part of the sort of doublet filtering is if you have too many UMIs per cell, because that's a sign that you might have multiple cells in it, or yeah, multiple cells in a droplet. Yeah, I think that's all the interesting stuff in here. And the other tabs, like for the most part, like not super interesting. It kind of does a little bit of analysis that you're going to ultimately want to do downstream, but it's doing it. It just does this sort of like default thing. So it doesn't know anything about like what the data is. So it's, you know, kind of clustering cells, like here, you know, you got like 7 different clusters. You'll talk about that some next week. It tells you like the top number, the top genes by, you know, differential expression across clusters. But of course, these clusters are, yeah, I mean, there's nothing wrong with it. It's just like not informed by the domain. Oh, here's where it's getting the sequencing saturation number. So actually looking at this, it might be, maybe I'm right. I don't know. It actually might be doing what's called the collector's curve if I'm looking at this. Anyway, yeah, so that's the report. Like I said, it's sort of like summary information. You kind of like, this report seems like totally fine. You do want to kind of like know, like here it's you know, it's telling us that we have, this is probably the most important number here, that the estimated number of cells. If you were targeting, you know, you have a control, I showed you that figure before, so you have sort of some control over how many cells you're going to get per sample. So if you were targeting 10,000 cells and you only got 2,300, then that might indicate a problem. And, you know, vice versa, if you were targeting like 2,000 cells and you're getting 10,000 cells, then that might indicate a problem. So these are kind of basic, and again, the number, like the median number of genes per cell, that's about normal, right? About 2,000, 3,000 genes per cell. And I can't remember what a normal number, I think about 10,000 reads per cell sounds about right. Sorry, 10,000, what am I saying? 10,000, average of 10,000 reads per gene seems about right to me. Questions? And like if this looks pathological, if it doesn't look like this, then you might have problems. Any questions before we go back and look? Okay. So so this opened another tab for me, so I'm just flipping back here, and we'll look really quick at how scRNA was run. Like I said, this chunk here at the top is just setting it up so that each of the subsequent chunks gets timed. Here that's weird. Can you see the gray? I'll switch to visual mode. Uh-oh. Let's see. Okay, good. We'll do that. That might be better. Maybe not. Okay. So we're using dplyr. Have we covered, I think we've covered like sourcing the config file, right? Anyone need me to go over that? Okay. We're just making an output directory for scRNA to put the output files in. This is just telling us, that's that little bit that told us where that output was going, which made it easy to copy and paste it to open in the file browser. And then this is the command that's kind of doing everything. I'm calling scRNA. I don't know if you noticed, but it printed the number of CPUs and the amount of RAM that was being used, and which library, that's specified in the config file. And so this is the command to run scRNA, right? And it's this command scRNA count. We have to, let me see. It's one of these matters. One of these is allowing, I can't remember which it is. So either ID or sample. For both of them, we're putting library. One of them will be used to like name the output files and like label here, like where it tells you that's the sample. One of them is telling it, so that this directory here, this -- fastqs, is telling it the directory that the input fastq files is in. And there's actually all the fastqs for all the samples are in there. and one of these, I can't remember which, is telling it which sample to actually, it should run on, because Cell Ranger, you can only run on one sample at a time. This is telling it the reference genome, the 10x like official processed reference genome to use. This is telling it how many CPUs to use, which is also specified in the config file. This is telling it how much memory it can use. And this is telling it how much virtual memory, which we always set it the same. I can't remember what the difference is. I think that if you have a small amount of memory, then you might set these different. But on the cluster, we have more than enough, so we set it for 64 for both. And then here's where I couldn't remember if we had to specify which library prep kit we used or if it figures out. And in fact, this is where we're specifying the library prep kit. And there's some place, I can't remember where these are like the nicknames. There's some table that 10x has where you can like look up the sort of abbreviations for the library prep kit. And that's it. It's, like I said, it's a black box. So you kind of give it the fast keys, you give it a little bit of configuration information. There are some other command line options here, but it does a lot of stuff under the hood that you don't have any control over. So it's actually kind of boring. Questions? So we've got 17 minutes left. I'm going to try and stop at 3:50 so that you have a little bit of time to fill out the survey. We have two options for the next, say, 7 minutes, 6 minutes. We can talk about the answer to my question about how you get the all the cell barcodes on a given bead to be the same. Or I can talk a little bit about, I told you that this isn't exactly how we would normally run Cell Ranger on the cluster. Can tell you a little bit more about how we would actually really do it. Anyone have a preference?
The first one seems more interesting, like
Kind of, although I'm not gonna, it's a little bit, I mean, maybe there's time to do both, but. So which one are you voting for?
I vote for the first one.
Anyone else? The first one is actually like really simple when I tell you, I don't think I figured it out myself. I think I had to look it up.
Is it bridge amplification?
Nope. It's even dumber than that. So yeah, so the question is, how do you get, I told you every bead has the same cell barcode on it, right? It has to have, for this to work, for a given bead, all the oligos on the bead have to have the same cell barcode. So all the oligos have different UMIs. That's easy. Well, I think it's easy, but I can explain. So this is actually really simple, and it has a little bit to do with what we talked about for the like basic intro to high throughput sequencing, how that works. So you take a bunch of beads that don't have any oligos on them. And of course, now I have to remember which way well, we'll ignore the direction. It's easy to get the R1s all the same, right? Because that needs to be, across all the beads, needs to be identical. So let's say you've got the R1s already made, and you get to so you got this bit made. Now you want to make the 10x barcode, and you want all the oligos on the beads to be the same. So you take your beads, let's say you have like, I don't know, we'll say it in grams. Let's say you have 10 grams of beads. So you take, actually, let's make the math easier. Let's say you have 40 grams of beads. So you divide the beads up into 4 different groups of 10 grams each. And you put those beads, each of those 10-gram things of beads, into a different tube or beaker or whatever. And you put the ATP analog in one, you put the GTP analog in the next beaker, you put the CTP analog in the next beaker, and the GTP analog in the next beaker. And I'm saying analog because they have terminators, just like for the Illumina sequencing. And so only one base can be added. And you're doing this chemically, you don't do it with a polymerase, right? So you have to like be a nucleotide chemist to like know how to do it, but that's been figured out. And so in the A beaker, A's get added to all the oligos on all the beads. So you let that reaction go, then you take all your beads, and assuming you basically want it random, there are different ways you could do it, but assuming you just want it random, take all the beads, you put them together, you kind of mix them up, you divide it into 4 equal parts, and in one beaker, you put in the ATP analog, in one beaker, you put in the CTP analog, in one beaker, you put in the GTP analog, in one beaker, you put in the CTP analog. So all the oligos in the A beaker get an A added. So all the oligos on one bead get an A added. And just keep on doing that for, what did I say, 16 base pairs? Yeah, so you do that 16 times, and then you have, now you've made sure that you added the same base on all the oligos on a given bead. And then like for the UMI, I told you the UMI is easy, but basically you take it all together and then you just throw, now you have all your beads. You don't care what goes on anything. So you just throw ATP, CTP, GTP, TTP all in there. And I skipped the step. You actually have to, somewhere in there, you have to like take off the terminator so you can add the next step. That's how, like, it's the same way that you, like, if you're just synthesizing oligos for like where you have one tube, you have, 50 base per oligo. That's also done with a terminator that's reversible. So yeah, it's really simple until you, like, of course, now that you know, it's really simple. Does that make sense? Questions about that? Okay. I got like 30 seconds, so I'll tell you just really quickly, just so you have a little bit of an idea. So the way we would normally do it, we'd basically use this script, and if you want to dig in, you can look, let's see, where is it? If you click on the R cube, we're going to go back to Assays, single cell RNA-seq. It's in this prep directory. There's actually a slightly different version of that R markdown file that I just showed you. These files are for downloading the data and the metadata. And I think it's this one. So there's a mechanism, the software that runs on the cluster so here we're using the RStudio interface, but if you want to run jobs on the cluster, you have to use a system called slurm. It's called a job management system, because there are thousands of computers in the cluster. Thousands? I think that's right. Hundreds at least. And like different people have different access to different computers and stuff like that. And so slurm is basically, it's kind of like when you, I don't know, you go to the bank and you like take a number for your return. So it's similar idea. And it has several different mechanisms that are also like kind of stupider and simpler than they sound. And so you can basically start a group of processes where you want to run the same pipeline on a bunch of different input files. And so that's what we do. Basically, for each of the input FASTQs, or each of the input samples, this R markdown gets run on a different file. And it actually lets you, makes it really easy to like say like, okay, here are the files, here's the program I want to run on it, just like do that. And so it can run it on multiple separate computers independently. So there's some details here that, and actually here it's even downloading the raw data before it's doing that. This is the business end where it's actually running, it's actually saying, okay, run this R-mark. It's actually running another script that's actually running the R-markdown on each of the separate samples. So it makes it faster. Oftentimes, I don't bother to do this because oftentimes I'm not really in that much of a hurry. But for this data set, actually this data set we probably could have gotten away with not doing that, but it does make it a little faster. That's the like 9,000 foot view. And I will say that getting this to work right can be a little bit of a pain in the butt, which is part of the reason why, like this data set, probably if we ran it just like sort of directly, just like ran it on one sample, then the next sample, the next sample, you know, maybe it would have taken a week, half a week or something like that. And actually, as far as I'm concerned, I'd rather let it run for a week and come back than spend a couple of hours of my time getting it so that it runs in a day instead of a week. Okay. Any other questions? If not, I'm going to, wait, where's the other? I'm going to put this back up. Please do the survey, and otherwise you will be going, and you don't need to, if you didn't run the cell ranger on that sample, it's okay. We'll have like the pre-processed data all ready for you for next week.